Hello, this is the video and we're going to take you through the genetic mutations tutorial. Um, Before you get started, I want you guys to notice what I'm going to do here. I never do this. Wait, first we're going to go over the objective. In this lesson, you will make and defend a claim based on evidence that inheritable genetic variations may result from genetic mutations. What we're really going to do here is we're going to talk about how genetic mutations can cause changes in the DNA and create brand new traits never before seen. And this can lead to several things, good and bad. This is what I never do before, is go over the tutorial contents with you. So there's gene regulations, mutations, there's an activity, science in the news, and a summary. I never go over the summary, and I'm not going to. But the bulk of what I'm going to talk about today is mutations, because this first part, gene regulation, I got to tell you guys, this is some pretty advanced stuff, and I'm going to go over it, but I've even linked a few videos for you to help you understand it, but it's pretty in depth, and I don't want you to quit on me. I want you to give it a shot, but understand that from as we go further, um, the hopefully the questions on the test will come straight out of the notes, and that's just what you need to do. Is just go straight from the notes, okay, and um, and try to answer the questions, because this is stuff I learned my second year of biochemistry, my fourth year of college, and I have no idea where they're teaching it to you, but I'm going to teach it because I find it very interesting. But mostly, this thing is going to be about the mutations, the lesson, and the science in the news. Let's talk about the previous question. I can't show you the previous question because as the teacher, it always gives me the answer, and I already know the answer. But the question was, are genetic mutations always bad? And so, first of all, let's take a look at that question. Are genetic mutations always bad? When you hear the word always, you got to think to yourself, wait a minute, that means every single genetic mutation every time is bad. And is there a chance that a genetic mutation can be good? I hope y'all didn't hear that beep, but it's okay. Well, the truth is that occasionally genetic mutations are good. And so as a result, that means that that question is false. So let's talk about this question. The white wallaby, that's kind of like a kangaroo, in this image has a mutation that gives it a white coloring. How could this coloring affect its survival in its environment? I'm not going to give you the answer to this, but I want you to think about this. They're brown. Are they brown just by accident? Is it random? Or are they brown for a reason? And if they're brown for a reason, what happens to the white one? It should be pretty easy for you to get that answer. Just write it down, please. Okay, this particular page isn't too tough. This is about what we call gene regulation. This whole thing is going to be on regulating gene expression. What do we mean by gene expression? Well, information in the chromosome, actually in the DNA that makes up the chromosome, if you'll remember that from last week. Chromosomes are made up of the DNA, and in the DNA there are slices of it that are genes. Genes contain the information for making proteins. That's called gene expression. To get the information from a gene and turn it into a protein which builds your bodies, okay, requires a two-step process called transcription and translation. Gene regulation is the ability to control which one of these genes at any particular time are transcribed and then turned into proteins. It happens differently in prokaryotes and eukaryotes. Prokaryotes are the bacteria, eukaryotes are literally everything else from fungus all the way up to humans. Now, let's keep this in mind. You have 23 chromosomes, and inside of each chromosome is a bunch of DNA, and along each of these pieces of DNA, there are some blank spaces like the red spaces here, and then there are some spaces that hold information like the green for gene A and the blue for gene B. Which one of these gets expressed is what we're talking about here. Which one of these where the information gets copied and turned into a protein through transcription and translation, that's what we're talking about. So here is how it's done in bacteria or prokaryotes. It's very simple, okay? Here is the actual the gene where it says ZYA. That's the gene. It's going to be longer than that, but let's just pretend. And this is where we get the information for the protein. But in order to determine whether or not it has to be expressed or used, depending on the needs of the cell, we have a whole structure called an operon, which includes the gene and the on-off switch or the operator and the promoter. And that's where the enzyme for polymerase, that's where it starts. This is basically where it starts. It attaches here and it says, okay, this is the beginning of the gene. The operator says yes, go, or don't go, and then it copies it or not, depending on the operator. This is the operon. This whole structure from here all the way to the beginning of this is called the lac promoter or the lac operon. Okay. Now, outside of that is a regulator gene, and it plays a special role, or it plays a role. It codes for the control of the operon expression. Because the transcription gene, because the regulator gene is not transcribed, it's not part of the lac operon. But it's all of this right here is how do we decide to actually start copying this gene? Because this gene makes a protein that we may or may not need. We may need it, we may not. That's what this white one does, okay? This right here, the regulator gene, determines whether or not we need it. The lac operon is where we start, and then the operator says go, okay? And then it starts copying it down through here. It's like this is the beginning of the track. We put the train on the track. The train goes down here and makes a copy of this DNA through transcription. But the regulator gene is the one that says, okay, this is where we're going to decide whether we need it or not. Now, I know that's a little confusing, but like I said, just stick with us. And so here's another example of it. This is a different operon. This is the TRP operon, TRP operon. Here's the promoter, okay? And this RNA polymerase isn't always there. But here's the promoter we talked about just a minute ago. Here's the actual gene right here, okay? And then the regulatory gene that's outside of the operon is somewhere else on the DNA, okay? In this case, it's further away. But this is the one that says, okay, we need some of this stuff. This gene makes something called tryptophan. Tryptophan is a protein that almost every living thing needs. You get it from the environment. You get it in your diet. But if you don't get it, then you have to make it. And so this regulatory gene right here says, okay, do we need it or we don't need it? If we need it, then it says, okay, make this stuff right here called polymerase and start copying it. That's what happens. And as complex as it sounds, it's really not that difficult. Here is the information. Here is the promoter. This is where the RNA polymerase, which is the beginning of the process, attaches, and it's going to work its way this way, making a copy. This over here, though, is the thing that says work or don't work. And this inactive repressor, it helps it to make the determination. If this thing if this thing creates this, it means go. And it only creates it because we don't have enough tryptophan. If you have enough tryptophan, this thing doesn't say make this and go, okay? So, all right, that's easy as mud, right? And looking back, I think I just told you wrong. Actually, tryptophan attaches to this and stops it. So if there's enough tryptophan, this thing says don't go, okay? That's what this inactive repressor does. If there's not enough tryptophan, this thing is removed, and that means start. So it's kind of like, imagine this being a switch, go, okay? This is a cover over it. If there's enough tryptophan, this cover stays over it and we can't flip the switch. If there's not enough tryptophan, this cover comes off and then boom, goes. So there's kind of a, there's tryptophan around here. This stays over that switch and it can't start. If there's not enough tryptophan, then this comes off and we start making tryptophan. And that's exactly what this explains here. This repressor is what I was just showing you. Okay, that's that other thing on the last page. It sits over the operator part of the gene, and it protects it. You can't start it if there's enough tryptophan. If there's not enough tryptophan, then that thing comes off, and then we start making our own tryptophan. It's crazy, but it's really amazing because think about it. Here's the problem. I don't need a ton of tryptophan. I only need to make tryptophan if I don't have enough in my diet. Well, this is keeping my genes from making tryptophan all day long because I don't want to make it. So this is the little piece of a cover over this that says don't make tryptophan until I say start. And so this right here, this repressor sitting on top of this operator says don't start as long as there's tryptophan attached to it. Okay? That's how these things are expressed. That's how all genes are expressed. This happens in your body like crazy as well. It's a little different, but it happens in your body too. Remember, we're in a bacteria right now. You're not a bacteria. So here's another operon called the lac operon. The lac operon contains genes that allow you to break down lactose. It's a sugar found in milk. We're going to actually create a gene called lactase, or I'm sorry, an enzyme or a protein called lactase. And if you've ever heard of people that are lactose intolerant, they don't make that gene. Okay. So again, here we go. The lac operon. We got a regulatory gene. Sometimes it's attached right next to it. Sometimes it's somewhere else, but there it is. Okay. If I've got the repressor sitting there, it says we're not going to start. See, the RNA polymerase can't even attach to it. We're not going to start because the repressor's there because I got enough lactase, right? But if I don't have enough lactase, this thing right here pops out, pink, and we start making a copy. That's really all it is. It's all about this little guy right here. And this whole thing is all about we start or we don't. Okay? And if that repressor is plugged into the operator, we don't make that gene. We don't make that protein. If because there's not enough of whatever the protein is that we're trying to make, this thing comes loose, and then we start making it until there's enough to say, okay, stop. It's just like a switch. That's really all it is. It's actually very incredibly amazing, but it's how we start and stop making genes. Okay. And this just explains it more. And I got to tell you, it gets into whether or not there's a glucose present and the allolactose and all that fun stuff. And I just don't think you guys need to know that, so I'm skipping it. And if it's on the test, then I will just give you credit for that answer. And as much as I'd like to see you be able to do this, just skip it. This is more than I want you to have to worry with. Don't fight trying to figure this thing out. Let's just go on. So everything that I just showed you is what happens in bacteria, but everything else is not bacteria, okay? And I mean, that's almost everything you can think of, although there's a ton of bacteria in the world, but that's a different story. Everything else goes through a slightly different process. So when we talk about regulating the process of taking the information from DNA and turning it into a protein, in eukaryotes, it's a little different. In fact, there are 4 spots where we can regulate this gene regulation, this gene expression, okay? So the first one is we do something about what we call the transcription. That is taking the information from here and turning it into a piece of RNA. That's called transcription, okay? We can control that. The second one is the process of the pre-mRNA splicing. That happens right here, okay? That's where we go from this to this. We can regulate that with some fun, fancy stuff, okay? From the mRNA degradation, it can actually break down and not be used here. That's not very common, so we don't talk about it a bunch. And then finally, the last part is taking the information from the mRNA and turning it into a chain of amino acids, which then folds into a protein. Now this process is really complex, and remember me telling you that it's, yeah, there's a video on it. I'd like for you to watch that. I'm not going to play it right now because that takes up time, and I'm not going to do that. You can watch it on your own. But watch that video. It does a very good job of explaining it. Now, in complex organisms, the eukaryotes, there's all kinds of things. The one thing we want to talk about that controls the transcription. That's the first type of control we talked about. It's called a Hox gene. Hox genes are really interesting, okay? Because Hox genes are a group of genes that regulate the formation of various body parts. Notice the color coding on this bug, okay? Let's just say the front legs, for example, the blue. The Hox genes, they code for very important factors, okay? And they determine which genes are expressed and how much, okay? The transcription of Hox genes therefore determines the body plan of an organism. It's kind of crazy. Hox genes are found in clusters and can be spread over all kinds of different chromosomes. We have 39, so the Hox genes are located. Um, we have 39 Hox genes in 4 clusters on 4 chromosomes. The fruit fly has 8 Hox genes, and it's spread out over 4 chromosomes, okay, in 2 complexes. Yeah, we don't need to learn all that. It's interesting though. This front 2 sets of legs right here on this bug, the Hox gene that says put those legs there is the exact same Hox gene that tells me to put my arms on my body. It's the exact same gene. So as my body is being developed, the same gene showed up right there for me as showed up for a bug, suggesting that there is some kind of connection between these 2 organisms, which is kind of scary. Okay, so this is the hardest slide you have to look at for the entire thing. Look, I'm going to try to break this down a little simply for you, okay? This structure right here is called a a transfer RNA. It puts an amino acid at the top off of reading the mRNA at the bottom, okay? We can, well, naturally this is messed up. This stops. It's called mRNA degradation and translation. And we can stop this by doing some funny stuff, okay? The only reason you need to know this is because this last part. Geneticists are exploiting this mechanism by introducing synthetic molecules who target these genes, okay? This means that if there's a gene that's causing cancer, we can figure out how to make this process happen artificially to stop cancer. That's why you need to know this slide, okay? We can also treat things like diabetes and even other diseases because any disease that's genetic goes through this process. And if we can stop this process right here from happening at the specific time, okay, then we got it. We're able to cure cancer. Here's the thing, though. We want this process to happen 99.9% of the time. It's required, okay? But we know how we can tell specific times for it to stop, and we're working on that. So that's one of the ways we might fight cancer. It's crazy. There's some very smart people, way smarter than me, figuring this thing out. Okay, don't get caught up in reading about enzyme dicers and RNAi mechanism through dsRNA. Don't get caught up in that. Okay, just understand that this is a normal thing that happens in our body, but it also happens in things like cancer. And if we can stop this from happening in the cancer cells, then we've cured cancer. That's what you need to know. I think you could answer this question, and I'd like for you to give it a shot. Okay, now for the fun stuff. Mutation is a change in the DNA. Okay, remember the DNA is like a series of letters. Okay, and if you read a book, all you're reading is a series of letters, right? What if I change one of those letters? Would it change the meaning of the book? Of course. What if I change one of the letters in the DNA of an organism? Would it change how that organism works? Of course. And the more mutations that occur, the more likely there are going to be changes. Let's be clear about some things. Most mutations are bad. They lead to the death of the cell. If they don't lead to the death of the cell, they usually lead to cancer, and that's bad, right? Occasionally, the mutation is in the right place at the right time and can actually, well, you know what? I want to back up a minute. Most mutations are silent, meaning that they don't do anything. You can't even tell. You're mutated. It's happened all the time. But it's silent and doesn't do anything because it's on a part of the DNA that's blank and your body doesn't use. Or, oddly enough, they changed the letter but that doesn't have any effect. Okay? But of the mutations that aren't silent, most of them are deadly. They kill the cell. Or worse, they create cancer. If they're not deadly and they don't kill the cell, they could happen in the right place at the right time. It has to happen in the sex cells. But it can actually create a change in the DNA that is passed on to the offspring, and then we get something weird. Now, this happens more often than you think, just so you know how often mutations occur. Okay. Mutations occur randomly, but chemicals and other substances in the environment can aggravate that Think of all the things that you've heard about that can cause cancer. Well, they also can cause mutations. Okay. In fact, cancer is just a mutation. Okay. Substances that can trigger mutations are called mutagens. Now, there's a class of mutagens that specifically cause cancer called carcinogens, but they're mutagens. Um, and so let's examine the different ways in which a strand of DNA can mutate. There's a game that goes along with this that I've linked into the calendar this week that I really think you guys would enjoy. The first type of mutation is called a substitution. Okay. Um, and basically what happens is one nucleotide is changed for another, and it changes then the codons that tell what to express. Now you remember we talked about codons last week. You got all these amino acids, right? There's only 20 amino acids, but there's 64 codons, which means that some codons code for the same amino acid. Now I know this is hard to follow along, but if I got a codon, if right here this is a codon that says make an amino acid or grab this amino acid, sometimes if I change that, it changes it to another codon that still says grab the same amino acid. That's what makes it silent, okay? And so if I accidentally swap out one of those letters, I'm going to change the codon. And if the codon is changed enough to get a different amino acid, then it changes everything. But if I change the letter but it still says get the same amino acid, it's a mutation, but it's a silent mutation, okay? But substitution is very simply this. I've got GGACA. I changed it to GGAGA. See how I changed the C to a G? That's called a substitution, okay? The next one's called a deletion. Quite simply put, that letter that's there, it just gets deleted for whatever reason. It gets knocked out of there. Bang, it's gone. Now that changes everything, because imagine if you're reading a book and all of a sudden a letter is deleted from a word. Does it change the word? Sometimes, or most of the time, it just changes the word to nonsense, and then all of a sudden the sentence doesn't make any sense, and then it's all done, right? Sometimes we can change the word to a different word, and then it totally changes the sentence. That's called a deletion. A duplication is when I make a copy of words that are already there, of letters that are already there. So imagine if you're reading and all of a sudden there's extra words that just got copied. Okay, that's called a duplication. It's best to just look right here. See this? This is what's normal, and all of a sudden that got added on. It's an accident, but it got added on. Now look at the DNA. It's different. Instead of A B C D E F G, it's A B C D E F G F G. That changes everything. Okay, creates a whole different protein, which creates a whole different situation. The next one's called an insertion. Okay, part of one gene is actually inserted into another. And so instead of having HILM, I now have HILM ABC. Totally different gene, totally different protein, totally different thing. And so insertions, you can see what happens. A little piece of gene from one place gets added to another one, and now I've got a whole different problem. Okay. An inversion, notice what happens. They get flipped. A portion of the DNA reverses itself. That's normal A B C D E F G, but what if it goes A B D C E F G? Now see it's how flipped and mutated. That's an entirely different sequence now. And if it were letters in a book, then it's an entirely different, means it has an entirely different meaning, which means it creates an entirely different protein, um, which has an effect. And then finally, there's translocation. Sections of DNA from two different spots swap. Okay, and now you just get a whole new setup here. I mean, see what happens. Parts of this one goes here, and parts of this one go here, and I'm totally goofed up. Okay, so that's called translocation. Okay, so this is the next question, and I'm covering over the answers with just my media player. So just you see what I'm doing there, right? So don't get confused. But basically, if you have an ATGC as the original, here's what it's asking. If the original is ATGC and then it just goes to ATC, what happened? Of all those things we just went through, is that an insertion, a deletion, a substitution, all those things? All right, you know what? I'm going to go ahead and give you the first answer. What do you think it is? What do you think it is? Yeah, it's a deletion because the G is gone. So the answer here is deletion. So if I have ATGC and I go to ATTC, what do we call that? If I have ATTCGC and I go to ATTCGGC, what happened? Okay, you see what happened? There's an extra G in there, right? What do we call that? Keep going all the way down, okay, and label all of these. Um, and this is a pretty easy question if you'll just use the last page of your notes. Now, in a minute, we're going to talk about what we call somatic, S-O-M-A-T-I-C. Somatic cell mutations, okay, are ones that happen to your body cells. But inherited mutations come from one or both parents via the sperm or the egg, okay? Once it's inherited, these mutations exist in all the cells in the body. Think about it. Every cell in your body came from an original cell that was the fusing of an egg from your mom and a sperm from your dad got together. Now, if that egg or that sperm carried a mutated gene, this spot here called an allele, okay? Here's a normal allele, but this one got mutated and it was in your mom's egg, right? Now, if it was in your mom's regular body, it wouldn't be in her egg, okay? Because that's not how it works. And then it goes to the whole thing whether it usually dies or it's silent or sometimes it be cancer. But if it happened to the egg as the egg was forming, okay, and the mutation occurred right there, and then your dad's sperm fertilized it and that became you, now you're mutated. Here's the funny thing. It's an absolute guarantee that this happened. Mutations are that common. It's an absolute guarantee that this happened inside of your DNA. You have so much DNA, there's no way it did not mutate, but it's more than likely silent, okay? Only very rarely do you get a mutation. Very rarely do you get a mutation that shows up. Most of the time, if a mutation shows up and it's from somewhere important, then it kills the egg and the sperm and just dies. Really, 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 really rarely does a mutation not kill the organism and the child is born. Most mutations are bad, such as PKU. PKU is a mutation of the PA gene. This gene codes the enzyme that breaks down the amino acid phenylalanine. You have this amino acid phenylalanine in your body, and it's useful, but it builds up, and you have to have an enzyme that goes there and breaks it down. People with PKU don't make that enzyme. An enzyme's a protein. If you don't have the DNA right that makes that enzyme protein, then you can't break down phenylalanine and it builds up. And it builds up in the brain and it leads to mild to severe brain damage. In fact, quite often it affects the development of the child and most of the times they die. Okay. The good news is you have to get the mutated gene from both parents. So see how rare it is that a mutation shows up? They're there all the time, but to actually see it expressed is really rare. Another gene that causes breast cancer. There's actually two, BRCA1 and BRCA2, and they're inherited, right? You can inherit these genes, and just because you inherit these genes doesn't mean that you're going to have breast cancer. It just means that you have a good chance of it. It's what we call autosomal dominant, meaning you have to get both of the dominant trait. The dominant trait is the mutated trait, and you have to inherit both in order for it to set you up for cancer, although sadly you're more likely to inherit the dominant trait than the recessive. And you have to, if you inherit one, you have a greater chance of having developing breast cancer because these genes, they're not there to actually make breast cancer. That's not what they do. They have another job. When they're goofed up, then they don't fix their job. By them not doing their job, that's how they cause breast cancer. So this isn't a breast cancer gene. It's not like, oh my God, why would there be a gene that says, okay, you have breast cancer? That's not what's happened. What happens is this particular gene has a job to do that prevents breast cancer. And when it's mutated, then it can lead to breast cancer. Cystic fibrosis is a mutation of the CFTR gene. CFTR in this case is autosomal recessive, which is the opposite of autosomal dominant. There's recessive. That means you have to inherit both genes, both mutated genes from both parents. Okay? So you get a mutated gene from mom and a mutated gene from dad. And this is really a Punnett square if you look at it. Here's mom and here's dad. They're both carriers. They don't have both traits, so they don't have cystic fibrosis. They have kids. There's a 25% chance a kid doesn't get a mutated trait at all. There's a 50% chance that the kid gets a mutated trait, but it's just a carrier, so they don't even have it. I know somebody that's this way. They're what we call an uninfected carrier. But sadly, if both parents carry cystic fibrosis, there's a 25% chance that their kids will get it. I know a person it's a female. This person has cystic fibrosis. They're a carrier. They don't show it. You would never know. okay? But they knew that they had it, and so before they chose to have a child with their husband, they had their husband tested to see if he was a carrier. And as a result, they found out that he was not a carrier, so there's a 0% chance of having an affected child. If they would have found out that he was a carrier, there would have been a 25% chance that they had the child that would have cystic fibrosis, and then they would have had a decision to make. And here's a decision for you to make. If there's a 25% chance that your child was going to have a debilitating disease that will cause serious problems. I mean, read what it does here. It produces a defective protein that causes a buildup of thick, sticky mucus. It affects several organs, mostly the lungs. It damages the airways. It can cause blockages in digestive organs, and ultimately it leads to death. There's a 25% chance that your kid was going to have that problem. Would you have kids? People make this decision. This is what we can do by studying DNA. This person found out that their husband was not a carrier, so they felt safe that there was simply no way that they were going to have a child with cystic fibrosis. At worst, there's a 50% chance that they were going to have a child that's a carrier. And now they found out. They let their child know that you may be a carrier. If you ever choose to have children, you should have yourself and your um, mate tested. So somatic mutations, somatic means body, soma means body, so a somatic cell is a body cell, it is not a sex cell. That's the only two things. You either have somatic cells or sex cells, okay? And the sex cells are created in the testes, or the organs, I'm sorry, the ovaries. So everything else that's not a sperm or an egg basically, or the tissue that creates the sperm or the egg, is a somatic cell, okay? So if there's a mutation in somatic, you're not born with it, so you have to acquire it. You get it through in your life through being exposed to mutagens such as chemicals, tobacco, sunlight. This is skin cancer. It's a mole that has turned cancerous. These cells are growing out of control, okay? It's caused by something that happened in this person's lifetime to those cells, probably really just one cell, and that cell's now growing and dividing and growing and dividing and turning into a dangerous cancer. So this is skin cancer. Okay. Um, it's only present in the type of cell that the mutation has occurred. Skin cells um, have skin cancer. This type of cancer is not going to be found in the bone, all right, because it's not bone cancer. It's skin cancer. Most cancers arise from somatic mutations. So when we're talking about cancer, we're talking about the result of a mutation that has caused the cell to go crazy. One cell goes crazy, and then all the daughter cells after that have the same mutation, so they're going crazy too. And it's like one turns into two, and they're crazy. Two turns into four, and they're all crazy. Four turns into eight, and now you got a family of crazy. And next thing you know, you get crazy, crazy, crazy cells, and it leads to this problem. Now, mutations aren't always bad. Well, let me rephrase this. Mutations don't always lead to cancer, especially in the sex cells. Okay, those are the ones that cause a rare mutation to occur in the entire organism. Um, this puppy on the right has more lightly colored um, uh, features than the normal features found on the puppy. Now that means that one of this puppy's parents had a mutated trait in the sex cell. Okay, and that was passed along to this offspring. And as this offspring grew, every cell that came after it, from that original merging of the sperm and the egg, every cell that came after that, which created this body, contains a mutated trait. Okay, and now all of a sudden you get a slightly different coloration. Is there any effect? Not really. I mean, it may. I could surely sit, if I had to, think of an environment in which this is a disadvantage and can cause this organism to struggle. But right now, that's nothing. It just makes a cute puppy. He looks a little different. That's a mutation. Most color mutations are neutral. That's what a neutral mutation is. It doesn't have a positive or a negative effect. It's just there, right? Sometimes color variations can cause a problem. It makes it where it has a worse chance of attracting a mate. Okay, believe it or not, dogs can see this and go, "Ooh, something's wrong with that." It can affect your ability to camouflage itself from predators. I'll go back to the original question with the kangaroos. The one was born white. Can you see how that would be easier for a predator to see? So therefore, it can't camouflage itself very well. So it's got a much greater chance of being killed in the wild. So sometimes they're negative. So we'll see. We'll talk more about them in just a second. Next slide. Great example of a harmful mutation here. I mean, take a look at this grasshopper. It's beautiful, right? But looking at this grasshopper up against that green, man, that thing sticks out like a laser beam, right? And so in this case, this mutated grasshopper has now got a far less greater, a less chance of surviving because a predator like a bird can see this much more easily. And so it's probably going to get eaten. Now, think about that. When you get a pink grasshopper, it's easier for it to be seen, so it's more likely going to be eaten, which means it's got a less likely chance of having offspring. It's probably going to be eaten before it reproduces or reproduces very much. And eventually, that trait's just going to disappear. It's why we don't see mutations, harmful mutations, all throughout a species, because they usually kill the organism before it can reproduce. So harmful mutations don't spread as much as positive mutations, just like this pink wouldn't. Now, here's an interesting thing. Suppose this grasshopper was, found a place that was predominantly pink and red tones. And I don't know what kind of place that would be, but just imagine. Now all of a sudden it's an advantage, right? See, mutations are only bad in certain places. Some places they may be good. I don't know if this thing moved into I don't know, paint store or something and it was all red. That's kind of stupid. But then it's kind of an advantage because it can hide better than a green grasshopper. So sometimes mutations seem bad, but they're really good. It just depends on the environment that they're in. Sometimes. Most of the time, mutations just kill you. Those beneficial mutations uh, can give you a leg up. And this is the weird ones. These are the ones that rare. They're so rare for a mutation to occur that doesn't kill the offspring. And it's not a negative mutation, but it's a positive mutation. But a positive mutation is what leads to evolution. Certain mutations give organisms attributes that increase their chances of survival. The process of natural selection favors such mutations, which then spread through the species. Now think about this. This is a speckled peppered moth on the top, right? Now, that's what color it normally was, but during the Industrial Revolution in Europe, smoke from factories kind of covered. See, this was hiding on trees covered in lichen. I don't know if you've ever seen the sides of a tree. It looks kind of speckled. So this was a camouflage to help hide it on the side of the tree. It was speckled just like the side of the tree, right? But the soot from the factory kind of covered over those trees, and now they're no longer speckled. They're black. Well, this guy now sticks out like a sore thumb, but a mutation occurred to make them black. Now, if it had occurred before the Industrial Revolution and the trees were still speckled, this thing would stand out like a sore thumb. It's a negative mutation. It's a harmful mutation because something's going to see that and go, look, man, I can see that bug. I'm going to get it and eat it. But because the environment changed, now all of a sudden, when this mutation occurred, it became a beneficial mutation, and now it can hide better. Okay, and so now the moths with black pigmentation have stuck around, and there's getting to be more and more of them. And for a while, that's all there were black moths. Now, the Industrial Revolution has gone away. We're now not putting so much smoke in the air. The trees are no longer covered in black soot, and now they're moving back towards this. Okay, now this is no longer a beneficial mutation. Oddly enough, mutations are important. Beneficial mutations are important because they create new genes. Okay, so through sexual reproduction, we get all these combinations of existing genes, and we try all these different combinations and look around how many different kinds of humans there are. But even in humans, we still all have, only certain genes to pick from. There's only so many eye colors, skin colors, hair colors, shapes of nose, shapes of this, shapes of that. And after a while, it's beneficial for a species to come up with new traits. And so if a mutation would ever occur in humans, I think we're going to talk about that in a minute. If a mutation were ever to occur in humans that would create a new trait, if that trait is beneficial, now all of a sudden, this trait is going to spread around through the upcoming generations of humans. Bacteria do this. Bacteria become resistant to antibiotics because they mutate. You know how the doctor says, if I give you a bottle of pills, you take them, and you start feeling better halfway through the bottle of pills, and you think, I'm going to quit. Long story short, that's creating what we call resistance in bacteria. Because imagine that you take a pill and it kills 99% of the bacteria. That's awesome, right? But what can you tell me about the 1% of the bacteria it didn't kill? Yeah, didn't kill. They're resistant to that bacteria or that antibiotic. And now that's the one left to grow. So now all of a sudden they grow back. Now you're sick again. And you're like, oh, doc, I don't feel good. You said you didn't take all your pills, did you? No, I didn't. Can you give me some more? If I give you the same pill, it won't do you any good because the ones that grew back, they're all resistant. Okay? So now we got to try a whole new antibiotic. And eventually we run out of antibiotics and we get something called, like with a very common bacteria that affects humans is called Staphylococcus aureus. We just call it Staph aureus. Staph aureus, that's what the SA right here stands for. Staph aureus is a bacteria that infects you and it can make you sick and not feel well. Okay? We have now gone through every single antibiotic that we know of to take care of Staphylococcus aureus. And there is a bacteria, a form of Staph aureus, that has become resistant to every single bacteria or antibiotic that we have, every single one. And it's called MRSA, methicillin, that's what the M stands for, methicillin resistant Staph aureus. It's resistant to every antibiotic all the way up to the strongest one we have called methicillin. And if you get MRSA or MRSA, then you got a problem because we ain't got nothing to give you for it. This is caused by the fact that we have bred this resistance over the years by using too much antibiotics. Here is another activity that you have to do to get credit for this tutorial. It's called Huntington's disease. And what you have to determine is what is happening with Huntington's disease. It tells you what it is, okay? It shows you how it's inherited. But taking a look at this, is it inherited by having the dominant trait or the recessive trait? So look at all the information and then determine, do you get it from inheriting the dominant or the recessive trait? And then, the rest of this is not that hard. Okay, I know there's a lot to answer, but it's not that hard. Okay. All right, the last thing. Because we're doing things like you've heard of 23andMe and these other DNA testing companies that you can spit into a cup and send it off and they'll test their DNA and they'll tell you all about it. We're getting very good at reading people's DNA. This stuff's only 20 years old. In 2000, they announced that they could finally start reading people's DNA. It would take weeks to read one DNA from one person, but they figured out they could do it. In 20 years, we can now do it in, I think it takes an hour. You spit in a cup, you send it off, they put it in a machine, and it can come back in an hour. What used to take 2 weeks, they can now do in an hour, and it's going to get even faster. So now we have an ability to look at a lot of people's DNA, and people do it all the time. And I don't think it's a bad idea, but every time you send your DNA into, like every time somebody sends their DNA into 23andMe, they're collecting more and more information about humans because they want to know, okay, so this came from you. You're from, you live in Florida, but you're, I don't know, you're Hispanic, for example, which means your family probably migrated from somewhere in Central or South America. And so they keep all this information on you. Then they take a look at the genes on you and they say, okay, well, that means that you probably came from, Guanajuato, Mexico. And it's amazing what they can tell about you. Plus, they can start looking at mutations. They can see if you have mutations. And they can read those mutations, which usually don't mean anything. They can tell a lot about our evolutionary history in humans. It is amazing what just reading everybody's DNA and um, turning it into a huge database and comparing. It's amazing what we're going to learn. Okay, watch this video on mutations in our genome and tell me why the narrator talks about certain things as successful mistakes. Tell me what's a successful mistake. And here's the whole thing about color blindness. And color blindness, you might want to Google it a little bit, but it's interesting because color blindness is a gene. It's a mutated gene, but it's not found on chromosomes 1 through 22. It's found on chromosome 23, which is the sex chromosome. And with humans, there's two sex chromosomes. There's an X and a Y. X, everybody has an X, but if you have two X's, you're a female, and if you have an X and a Y, you're a male. Interesting. Did you know that most of the time males get red-green color blindness? What does that mean? It means it's either on the X or the Y chromosome. I'm curious to see if you guys figure this out. Try to find the answer, okay? It's very interesting how it works. And that's it. This is a really challenging tutorial. Genetic mutations, if we were in a regular class, I would have taught this to you over the matter of literally weeks. But that's not how we're doing in virtual school, so you're going to be doing a lot of follow-up on your own. Look at the calendar that I've produced. There's a lot of supplementary material to help you out. I strongly encourage you that you look through the videos, the games, and the websites and take some time to study them. It's going to help you learn this better. Of course, if you need my help, you can Zoom with me any day. You can email me, text me, even call me. All right, good luck. Get to work.